Hello everyone and welcome back to Genomics for All episode 16. And this week we have World Cancer Day. This is Cancer Day. The theme is by the Union for International Control of Cancer, that's UICC, and the theme this year, 2025 World Cancer Day, is putting people at the front. And the theme, what is being decided by the International Committee is united by unique. So that is each person having a cancer story is unique, and as you all will appreciate that it's a deadly disease, and everyone who has gone through himself or herself or someone in the family, this is disastrous. So this disease, it's more and more important for us, all of us, to talk about cancer. More and more awareness is required, and especially in our series, because genomics plays an integral role in cancer care and cancer management, cancer research, cancer treatment. Everywhere you will see genomics. So it's more important for all of us to talk in this series, Genomics for All, about cancer and World Cancer Day. So in the last two episodes we did on cancer. One was on genomics for cancer, its applications in diagnosis, treatment, minimal residual disease, everything I had covered. And then we also talked about specifically on breast cancer, wherein we talked about the whole Pink October and applications of genomics in breast cancer. So today we'll shift gears, and I would like to bring to you all of your attention a very important topic, that when clinicians and scientists partner, I think a lot of very interesting, innovative research can come. And I urge all my clinician friends all over India to come together with scientists to work together. Like I would say, universities and medical colleges should work together to bring in new care, new treatments in cancer particularly, and many other areas. So in that, I would today I would like to talk about what my biome has to offer, and I would request all my clinician friends to look into all of these and see where we can work together. There are so many areas to work together. So this is the theme of today's Genomics for All video series. So what my biome does in the areas of cancer, I tried to categorize by different sample types that we work with, and that's easy for my clinician friends to understand. So in sample types, when I say we work with blood, and within blood we work on exome sequencing, which is the sequencing of all coding regions, 20,000 genes in human, and we work on hereditary and ovarian, breast cancer diagnosis using blood. So these are the two areas that we work in the blood sample type. Exome sequencing to know all your predispositions to cancer like BRCA1 and BRCA2, and a specific panel called HBOC, hereditary breast and ovarian cancer panel, where we work on specific genes for breast and ovarian cancer. So that is where we all can partner, work together to delve further into research as well as applications in diagnostics as well as therapeutics for this area. The next sample type, which is very important for my clinician friends, is formalin fixed paraffin embedded samples, FFPE as we popularly call it. So these are fixed samples wherein the surgery happens, and immediately these samples are paraffin embedded. So this is a very important sample type for us at my biome, and what we do from here is again, as I said, exome sequencing and panels. So this will be the focus of many sample types here that I will talk to in the next few minutes. So here what we do is exome sequencing, and we have panels like 52 gene panel, that is pan cancer, lung panel, that is 20 gene panel specific for lung cancer. Then we have a comprehensive genome profiling, which is a 500 gene panel, very interesting panel, and I urge all my clinician scientists to get in touch with our team to do more and more with this panel. We can do a lot using Indian data. This is a comprehensive genome profiling for 500 genes, all relevant cancer genes that have been studied till date. It's an amazing panel, FDA approved, etc. So that we have been doing lately. We do many other panels, but then bigger panels like exome sequencing, and then other studies on research using methylome, which is the epigenetic patterns in the cell, transcriptome, which is the expression pattern, RNA-seq that we call it. So this is another sample type. So we covered blood, we covered FFP, the most important two sample types. And the third factor is the liquid biopsy that I have been talking about in different talks in Genomics for All. And this has become very important because using liquid biopsy now you can detect cancers early. So early detection of cancer by liquid biopsy, which is the using the circulating tumor DNA or the cell-free DNA, not the blood cell DNA that I talked about just now. So using cell-free DNA, one can monitor early detection of cancer, manage cancer well by using markers, panels, exome, etc. to understand what is going on in the management of cancer, and minimal residual disease, whether relapse will happen, etc. So amazing applications of liquid biopsy are coming on, from early detection to management to minimal residual disease, all in one test called liquid biopsy. So we have been doing that at MyBiome, and I urge again all my clinician friends to discuss this potential opportunity with us to do more and more research on using Indian data on liquid biopsy in cancers. So that is the third sample type, and the other sample types belong to the reproductive area, wherein in female reproductive biology we have been working with human papillomavirus, as I talked about in one of the video series for cervical cancer. A lot of potential in this. Using cervical swabs, we are able to now detect 28 human papillomavirus subtypes, 13 sexually transmitted diseases, sexually transmitted infections, and more. So that is where we are using NGS, next gen sequencing, using our state of the art lab, having MySeq, NextSeq, NovaSeq, Oxford Nanopore, Promethean, Mina, and many platforms in our lab. We are able to detect all of these quite significantly. Then comes a different topic, that is vaginal microbiome that we use in our lab, and that recently has been attributed to the development and diagnosis of cancers. So again, a very interesting topic, very less has been worked upon in India at least, and very nice opportunity for us to partner using the excellent science that we are creating and the doctor community coming in. Apart from vaginal microbiome, there is another important aspect, that is the oral microbiome that we work with at MyBiome here in our lab, wherein it's very interesting to see that signatures of microbiome in your oral cavity, a simple saliva test or an oral swab will tell you about the development of oral cancer, and that's the beauty. Like a simple test, a swab test can tell you whether cancer is developing in your body. So that is where we are with technology today, and let's utilize all these technologies to go further with new tools and diagnostics for oral cancer. As in our company, our sister company Microbiome Research Private Limited, we always talk about gut microbiome and its applications in numerous areas like food, probiotics, prebiotics, the whole recommendations for different lifestyles, gut health index, etc., and more we talk about in the microbiome series. But here I would like to bring to attention one particular topic, that is gut microbiome and its usefulness as a marker for cancer. In colorectal cancer particularly, one can see signatures of gut microbiome and one can diagnose it quite early, and these are fantastic applications of NGS based products and services that are coming, and we have to make full utilization of this science to further apply it very quickly in medical science, clinical practice very soon. And the other areas that I would like to touch upon are tissue microbiome, wherein there is a microbiome that is resident inside the tissue, like colorectal cancer. Within the tissue itself there is a microbiome, and there are distinct signatures. We along with some of our partners in hospitals are working on this subject. So these are some of the other sample types, and then there are various other sample types like cerebral, spinal fluids, and many body fluids that we can work with to develop very good sensitive specific NGS diagnostic tools and more research into the therapeutics part, treatment applications for cancer. So with this, I would like to wind up and again call upon all my clinician friends to come together with scientists to develop new products, new services, new platforms, new treatments, and there is always hope. As I say, with more science there is more hope, and wishing everybody a World Cancer Day to bring in more awareness for cancer diagnostics and treatment. And please read more about cancer and the stories that cancer brings in. United by Unique is the theme for this year's World Cancer Day, and thank you all for supporting Genomics for All.